Okay, here is the review for the chapter 3, or the topic 3. Cell test. Robert Hooke, he looked at cork, and he coined the term cells, right? Because they reminded him of the cells of a monastery that the monks would stay in. Anton von Leeuwenhoek, he improved on the microscope, the compound microscope. Right? And he would make a lot of drawings of what we call microbes. And he would look at pond water and draw all the little animals that he saw in there. He referred them as tiny little animals. Matthias Schleiden was the German scientist who came up with the discovery that all plants were made up of cells. And Theodor Schwann was the German scientist that said all animals were made up of cells. Rudolf Virchow was the Russian physician that talked about how all cells come from pre-existing cells, which happens to be, right? All cells come from pre-existing cells, or all comes from other cells. One of the 3 components of the cell theory. The other one is that all living things are made of cells, which is pretty much what these 2 guys said, right? Plants and animals, but there's also fungi and prokaryotic organisms and protists as well. All living things are made of cells. And cells, what are they? Well, cells are the basic unit of life. The smallest you can get and still be considered alive is 1 cell. Prokaryotic cells, do they have a nucleus? No, they do not, because this is pro means before, karyo or refers to kernel or nucleus. Eukaryotic cells definitely do have it. They do not have membrane-bound organelles where eukaryotic cells do. Both types of cells are going to have cytoplasm. Prokaryotic cells are typically 1 cell large, so they're single-cellular or unicellular organisms, whereas eukaryotic cells have the ability to become multicellular. It doesn't mean that all eukaryotic cells are multicellular. A lot of protozoa are single-celled eukaryotic organisms, such as amoebas and parameciums and things like that. Both are considered a living thing, prokaryotic cells and eukaryotic cells. Both will actually have cell walls. They're of different composition, but you have prokaryotic cells contain cell walls made up of peptidoglycan, and eukaryotic cells both have cell walls as well. And they both have genetic material as well. They both have DNA inside of them. So how to plant animal cells? Well, they both have a nucleus, but animal cells do not have a cell wall made up of cellulose where plants do. They both definitely have to have a cell membrane, as do all cells have a cell membrane. Both of them also have a mitochondria. Oftentimes people think that plant cells don't have mitochondria, so like animal cells have mitochondria and plant cells have chloroplasts. That's not true. Mitochondria is needed in both animal and plants in order to break down the food and sugars in order to make ATP. Although animals, like you and I, right, we do not contain chloroplasts. Only plants have chloroplasts because it's in the chloroplast where this happens, photosynthesis. Right? And animals obviously don't photosynthesize. Both animals and plants need ribosomes. But only plant cells have a large central vacuole. Animal cells do have vacuoles, but they don't have that large central vacuole. Animal cells have centrioles, yes, whereas plant cells do not. And animal cells have lysosomes, whereas plant cells do not. Okay, and so that's unique to them. So if I was going to draw a cell membrane, I would first start with a bunch of phospholipid heads and their tails. Right, and so since phospholipids form a bilayer, here's my phospholipid bilayer. Okay, this is the lipid bilayer right there. these are the phospholipid bilayer. This is the lipid bilayer. Okay, lipid bilayer. And one of these guys is a phospholipid. Now, a protein would be like wedged inside. So this would be like a protein channel. And so here's your protein channel. And there. And on some proteins, you're going to have carbohydrates, right? So on some proteins, you'll have actually these carbohydrate chains that help identify and tag the cell. They act as identifying markers. So there's your carbohydrate chain right there. That's your carbohydrate chain. So an isotonic solution, if you have a beaker, right, of water, and you separate it with a membrane, an isotonic solution is going to be equal in concentration on both sides. A hypertonic solution, right? A hypertonic solution is going to have greater concentration, so the hypertonic solution is going to be that solution right there, whereas a hypotonic solution is going to be the one that has a lower concentration of solutes right there on either side of a membrane. So if I'm going to draw a typical plant cell that has all those structures and a typical animal cell, you're probably going to, the best bet is going to Google plant cell and find a good old-fashioned image of a plant cell. There you go. And look at all those structures, right? And then find a good old-fashioned animal cell, right? Same thing. Bam. And then draw all those structures. Notice the animal cell doesn't have a chloroplast. It doesn't have a central vacuole. It doesn't have a cell wall. Okay?